Good afternoon. Thank you for joining us for Research 411, a talk show focused on sharing best practices and trending research topics delivered by professional staff and prominent UT Dallas researchers from various disciplines. I'm Tiffany Willoughby, Research Education Programs and Outreach Manager in the Office of Research. I will moderate today's discussion. Joining me today to talk about stabilizing delicate drugs are Dr. Jeremiah Gaston Smith, Dr. Gabriel Meloni, and chemistry research assistants Fabian Castro, Nisansala Abirathna, and Samira Abirathna. Dr. Jeremiah Gaston Smith earned his Bachelor's of Science degree under the tutelage of Professor Joseph Gajowski at Indiana University, and his PhD from Professor Bradley D. Smith at the University of Notre Dame, where he studied the synthesis of sterically shielded near-infrared luminescent dyes. After his PhD, he traveled to Northwestern University to learn under Professor Sir J. Fraser Stoddard. At Northwestern, he investigated a diverse array of topics, including gas sequestration by cyclodextrin-based metal organic frameworks. He joined the faculty at the University of Texas at Dallas in August 2013. In 2015, he was awarded his school's highest teaching award. In 2016, he was awarded a new doctoral investigator award by the American Chemical Society's Petroleum Research Fund. And in 2017, he was awarded an NSF Career Award. Dr. Gabriel Meloni's research interests focus on the chemistry of metals in biological systems. Meloni holds a PhD in bioinorganic chemistry from the University of Zurich, Switzerland, and subsequently has been a Marie Curie postdoctoral fellow at the California Institute of Technology and Aarhus University, Denmark. Dr. Meloni's postdoctoral work investigated the chemistry and structural biology of transition metal transporters. He established an independent research group at the University of Texas at Dallas in 2015 to implement a research program that builds on his bioinorganic experience. His research group focuses on the development and application of biochemical and biophysical tools to characterize at the molecular level the structure, reactivity, and metal binding properties of soluble and transmembrane proteins and biomolecules involved in transition metal homeostasis. The research relies on a common bioinorganic chemistry denominator aiming at elucidating the mechanisms of transport, reactivity, and coordination chemistry of essential and toxic transition metals, as well as metal-based complexes utilized as anti-cancer drugs or metal-based compounds in diagnostics. Welcome, all of you, and thank you so much for joining us today. In layman's terms, can you tell us a little bit about your research, Dr. Gaston Smith, then Dr. Meloni?
Sure. Our research focuses in two distinct areas. We're really interested in preserving proteins and other therapeutic drugs and nanostructures that have a tendency to go bad very quickly. So they are very thermally unstable, or they're unstable to shear forces or vibrations and stuff like that. We're interested in preserving them so that they are robust and they can be stored at room temperature, and even more so that they can slowly release in the body. So think of things that like super long delivery of some therapeutics and drugs, and that's sort of where we're looking at.
Thank you. And Dr. Meloni?
Sure. Thank you, Tiffany. So as you mentioned in the nice introduction, we are interested in our lab to study biomolecules, in particular proteins that are present in every cell, in every organism, in particular proteins that are capable of binding metals. And this is particularly important because maybe we do not recognize very often that transition metals are actually essential elements. You can find those elements in like the pills that you take as supplements every morning when you take them, because these transition metals are actually performing very critical function in living organisms. Due to the fact that they are very reactive, the evolution basically evolved a lot of biomolecules that are in place to regulate the metabolism of these metals inside our bodies. And specifically in our group, we are really trying to understand from really the structural atomical point of view, what is the function and the structure of this biomolecule, and how do they work, and how cells basically are capable of managing the homeostasis of those transition metals. So we are essentially at the interface between inorganic chemistry of metals and biochemistry, and we try to bridge the two fields to really bring new knowledge about those systems in biology. Especially because some of them, they're actually, some metals can actually be utilized as well as therapeutic drugs. For example, platinum compounds as anti-cancer drugs. So we really want to understand the molecular mechanism that are controlling those metals inside our body.
Thank you, Dr. Meloni. Now, have you and Dr. Gaston Smith had the opportunity to collectively partner on research projects in the past? And then could you also tell us how you came together to work on stabilizing delicate drugs?
Sure. Myself and Dr. Gaston Smith are colleagues, but also good friends. Despite that, we actually did not really have a formal collaboration before we started this work a couple of years ago, though we had many exchanges throughout the time. So the way actually we came up doing this work actually is related to the fact that a large part of my research group is actually interested in studying so-called transmembrane metal transporters that are biomolecules that are located in every cell membrane, because every cell has a membrane that separates the outside from the inside. And those transporter molecule, transition metal transporters, they are actually involving in shuttling metal from one side to the other of this barrier. They are membrane proteins, so they are very hydrophobic. They are not water soluble. And due to those properties, it's actually very difficult to work with them. Many people in the field, they have developed technologies to actually study this protein. And typically this involves stabilization in detergent to extract them from the membrane, and then eventually try to reconstitute them back in artificial lipid membranes to study their properties. And this is very important because you can then really dissect mechanism of this protein. But these systems are actually metastable. They are very difficult to work with. So you have really to be careful when you are handling these biomaterials. And despite very valuable, the studies are somehow limited by this intrinsic instability. And knowing that downstairs, Dr. Gaston Smith is actually a pioneer and leader in the stabilization and the utilization of crystalline organic frameworks to stabilize biomolecules, we were having a coffee together, and I exposed to him my problem. He exposed to him what they are typically doing in his lab. So we said, wow, this is the perfect match. I think we should try to do something that no one else did before. And I think this is related to the fact that to really study the two aspects that are involved in this project, you really need to have two very diverse expertise. And I think we have been so fortunate that we have been basically very close in space, and we are very good friends. So we were basically in the perfect position to try to address this problem. And Dr. Gaston Smith can follow up more on this, on his aspect of this contribution.
Oh yeah, it was kind of a perfect fit, At the time, I didn't have an espresso machine in my office, so the only way I could get coffee was either from a Keurig, but that just kind of tastes like sewage, or I could go to Gabriel's office on the second floor, and he was just giving away the coffee. I mean, those Nespresso pods are expensive. But, the opportunist that I am, I saw it as an opportunity to go out there and get free Nespresso. And we chat all the time, but, this is a particular area where it was pretty clear that this is something that we could contribute, and you know, it was a great opportunity to work with you know, my friend and now collaborator, and yeah, we were just kind of surprised. I mean, I'm going to let the students really expand upon it. I think really what was the most surprising was how well it worked, how well the interaction worked, and how well the science actually worked. And because, as you know, Gabriel was saying, the stuff that they wanted to stabilize is unbelievably delicate. I mean, it is very fragile, you know. From a non-biologist, and I stress that I am not a biologist. I play one in the literature. These proteins that they were making and these liposomes were some of the most delicate things I'd ever seen. You know, when you're a chemist, you buy something, you put it on the shelf, and 40 years goes by, and it's like, well, the chemical's still there, and it's still just fine. Whereas these proteins, these transmembrane proteins in this liposome, you know, from what Sam and Nishi found, they go bad after 48 hours in the refrigerator, and you can't freeze them, right? Because if you freeze them, they thaw, they break. Like there's no way to really even use these things. You have to make them and then use them, and so that presented, you know, that was a challenge. And ultimately, what we found was we could stabilize them so much that you know, after the students did all their magic, and I sat in my office, I bought my own Nespresso so I didn't have to go up to the second floor, drinking my Nespressos and playing on Twitter. The students came in, and you know, I took some of these things that are so fragile they fall apart in the refrigerator. We encapsulated them, structurally solidified them, put them in a USPS envelope, and I mailed them to my mom in Rhode Island. And you know, she's a scientist, and so she was excited to try this. She got it, put it, I don't know what she did with it, but she mailed them back, and then they sat because the COVID hit. They sat like in the inbox for a month or two, like they sat forever. So this experiment ended up taking a very long time. Like they sat, and yeah, the trio of researchers there, that they opened it up, collected it, re-isolated it, and it was still good. Like you know, so that was pretty impressive, and that was really quite cool. That, you know, the research and personalities and the need all came together from a pretty diverse background of characters that resulted in something I think is really potentially very exciting.
Thank you, Dr. Gaston Smith and Dr. Meloni. So I understand that therapeutics such as the COVID-19 vaccines require refrigeration, and you just explained how, you know, you mailed something to your mother to test and then send back. So these vaccines, though, traditionally require refrigeration to maintain drug function. What was the impetus behind the efforts to bypass the expensive cold chain? And Fabian, did you want to go ahead and address that?
Definitely, I would be more than happy. So as you mentioned, this particular compounds can only function when preserved at extremely low temperatures. Okay, so the idea was to provide a suitable shell that could bypass the need of such low refrigeration while still be able to remove the shell pretty easily and retain its functionality. So as we were talking the other day, we think of a chocolate. This chocolate that I mean, it's very tasty whenever you keep it in the refrigerator, but if you put it inside of your car at the Texas weather, it'll melt and it will no longer be as tasty. So we wanted to provide this protection to keep your chocolate inside of your car, travel around Texas, remove this coating, and still enjoy your tasty chocolate without having to refrigerate it, if that kind of answers the question.
Absolutely, Fabian. And oddly enough, with all of the talk about Keurigs and espresso pods, I can kind of see the encapsulation and protection of coffee products and maintaining the quality of those through transport and distribution. So yes, thank you very much for sharing that with us. Samira, can you take a moment to share with the audience what a lipid protein is and a lipid bilayer?
Yes, Tiffany. If you think about cells, including the ones in our bodies, we have these essential barriers which separate the inner and outer environments of the cell. These cell membranes, the composition of them is made out of lipid molecules. Therefore, they are like greasy layers in the cell. In the cell. So these greasy layers does not let nutrients. Most of them are water soluble nutrients. So they do not let pass through the layer. So there are specialized molecules in this greasy layer called membrane transporters to achieve this. Those are called membrane transporters. So we do study them by taking them out, but the biggest problem that we face is their stability. So that is why we are very excited about this stabilization in the exoskeleton.
Thank you, Samira. Back to you, Fabian. Can you explain in simple language what an exoskeleton is and what it has to do specifically with the stabilization of a drug?
I think the easier example that I can think of right now is Iron Man. You think of Iron Man, which is just a simple individual, like anyone else, that can perform outstanding things, but without its suit, the guy cannot really do that much. So this protective shell is like having Tony Stark, suit on, go fly, save the world, and then once he's done, take the suit off and move on and be a regular human being, if that makes sense.
It absolutely does. You painted a very clear picture. Now, how was the exoskeleton created?
So as we can see from the figure in the PowerPoint, we have this pristine material, okay, with this, like Sam mentioned, this lipid particles with proteins in the bilayer, and we basically combined metal with a small organic molecule that whenever they interact with each other, they form this shell. The shell was formed around this lipid nanoparticles, as we can see here. We stressed the particles accordingly, and once we're done with the stressing, we removed the coating. Going back to the Iron Man example, we went there, flew, saved the world, took the shell off, and showed that Tony Stark was the same before and after putting the suit. That's basically what you guys can see in this figure.
Excellent. Thank you very much, Fabian. Nissan Salah, how has the pandemic impacted your ability to collaborate with your colleagues on this project?
Yeah, actually, we started this project back in 2019. That means before the pandemic started. So we had done a lot of experiments before the pandemic, and we still had a lot of critical experiments that we need to complete. And then when the pandemic came, the labs were closed down completely, and we couldn't do any of the bench work for like 3 months. And then even when after we returned to the lab, it was like in a shift basis because of the capacity was limited for the labs. So it was actually really challenging because the thing is, me and Sam, we worked with the membrane proteins, and then Fabian had to encapsulate them in the exoskeleton. So it was a lot of teamwork, and we had to like coordinate when we work like together. So it was really challenging, but anyway, we could do it with a lot of challenges.
Thank you, Nissan Salah. And can you also tell me how you coped with some of those additional challenges, what were some of the things that worked best for you in working with shifts? I'm imagining in my head, like communication was key. How did you in fact communicate, hey, this has taken place first, this is what needs to take place next, and when you are in, can you please follow up with this?
Yeah, of course. So we actually had a lot of virtual meetings so that we could plan how we proceed with the things. So the virtual platform was really helpful for us, like to coordinate the things.
Perfect. Thank you very much.
Yeah.
Have you had the opportunity, Dr. Gaston Smith and or Dr. Meloni, to collaborate with other researchers or company partners locally or nationally or even globally to test the validity of the transport? And Fabian, you could answer this as well. And if you have, can you tell us what the transportation looked like? And do you have a cost comparison, cold chain versus drug stabilization?
So, and as I'm just thinking out loud, the collaboration that we've had in terms of this project was done with some people in Austria. Very well-known individual in the metal organic framework field that stepped up and helped us characterize these materials in a magnificent way. Other than that, we have not tested this platform by virtue of sending these materials and having someone else characterize them. And let me just brief you into why this transmembrane proteins, proteoliposomes, are extremely hard to work with. You need labs like Dr. Meloni's that are experts in the field. Is not the sort of science that any technician can really perform. It's something quite difficult to do. So that's why we've found that as an obstacle in terms of just sending this and having someone characterize it, again, is not as simple. And just to highlight on the importance of how this team came up together and how people here at UTD, both, especially in the chemistry department, and I'm pretty sure in all the other departments, how great of the great people, the great scientists that we have here locally.
Absolutely. And did anyone else want to weigh in on that question? Dr. Meloni?
Sure, we'll be very happy to. I think besides the fact that it's pretty clear, and we reiterated a few times that, collaboration was really key to make this happen. I think that, one of the most important aspects that this research actually is bringing up is that, we actually started with the proof of principle studies. So we actually did not start it directly with liposome containing well-known drugs that are used in clinics or vaccine, but we really wanted to demonstrate that these delicate systems that are actually used either for drug delivery upon encapsulation in the liposome or as vaccines like the Moderna or Pfizer vaccine now in the COVID, but many, many other. They basically use principles in terms of structure that is very similar. All these drug delivery system and vaccine, they are based on liposome, lipid-based particles in which basically there are additional components that can be the drug or an antigen to perform its function. What we really wanted to show is that if we take a general, very unstable lipid particle containing biomolecule, this can be stabilized. Once we could demonstrate that, I think that there are many, many areas and many, many directions that can be taken and also picked up by other groups after being able to show this as a proof of principle. And this is where probably where collaborations will also become more and more predominant. Now that we have this technology, I'm pretty sure that in particular, Dr. Gaston Smith group will have the possibility to collaborate with many other people that are developing similar lipid-based particles, but for very different application. And I'm pretty sure that the generalization of this exoskeleton formation can be really extended to many other systems. And I think that that is really the power of the research that we did, and is where really the collaborative aspect will become very important.
Thank you very much. And Dr. Gaston Smith, did you want to weigh in as well?
Sure. So in the context of why it's important to remove these refrigeration costs, if you look at if you conceptually think of a vaccine or vaccine distribution, the material has to be made in a facility that is low temperature, and then it gets shipped out in a refrigerated truck, and it has to go to a refrigerated warehouse where it's picked up, shipped in a refrigerated compartment in an airplane, arrives someplace refrigerated, and then it's refrigerated all the way until it gets to the clinic. And that involves not just the cost of the refrigerator, but also the forms and certifications, the personnel required to make sure the refrigeration is maintained throughout the entire thing, certification processes to ensure that the material arrives appropriately, training and handling. It also involves an enormous amount of waste. So upwards of 50% of vaccines can be lost due to issues in this cold chain infrastructure. And so an estimate that's been, so you'll find in the literature, is that the actual cost of a vaccine, the material cost, about 80% of it is wrapped up in this process necessary to keep it all cold. And that's really quite tremendous. And if you put it in the context of COVID vaccines, the COVID vaccines are not per injection, are not that pricey. It's estimated a couple of bucks per person on Earth, but 37 cents of that injection is just to keep it cold. That's just to keep it cold. That has nothing to do with the amount of material that's wasted because things are left at room temperature, which is obviously going to be extremely high. And this does not include the cost of that certification process and all that. You start compounding these up, and then they become extremely expensive. And then to top it off, you have areas and this isn't just the really delicate lipid membranes. This is also the virus-based or vector-based and inactivated proteins. All of these sorts of things require this sort of refrigeration. And then you have huge areas that have millions and hundreds of millions of people in India and in North Africa that do not have constant power, right? They depend on solar generators for refrigeration, which obviously work well enough if you can bank those in batteries, and then they don't work if there's no sun. And so then you have infrastructure issues of actually driving refrigerated trucks and refrigerated buggies and motorcycles to these locations. And all of these things present not just a fiscal issue, but a humanitarian issue as well. Is that, are these people not going to be vaccinated? Or what would that even look like? It's been a long time since the WHO went out in the field and tried to vaccinate people against smallpox and polio. That was a worldwide endeavor on the level of the Apollo program, right? Like, it was a massive effort, and not just in convincing people to get vaccinated, but getting the vaccines to people. So I think that there's a lot of interest in developing these sorts of things for vaccine technologies, and we have struck collaborators at the University of Colorado, the University of Texas at Southwestern, and now in Sydney, Australia, that are interested in developing these for various types of not only just vaccine storage candidates, but what's really interesting, in fact, we have a collaborator here at UT Dallas and at the Pasteur Institute. What's really interesting is that this material not only protects the membranes and the surface, but it's actually completely biodegradable. It's safe. You can directly utilize this. You don't have to remove this surface if you don't want to, although that's a great way to store and stockpile these sorts of things, but you can directly inject it. And we believe that is a way to not only protect the material, prevent it from being refrigerated, but we have, and we should hopefully have a publication on this pretty soon, a publication or two on this pretty soon, that it actually enhances the immune response because it slowly biodegrades, and that produces a much stronger immune response. So I think, the transportation issue and cold chain remediation are very important, but I think this has a lot of legs, and we're very excited to see where it's going to go.
Absolutely, Dr. Gaston Smith. Thank you so much for sharing that additional information. Now, the vaccine. Is it preserved longer in the cold or with the exoskeleton? In perfect cold conditions, right? If you're able to keep it cold and you're able to keep it in an exoskeleton, do you know which one you're able to preserve in longer?
We are currently collaborating with some labs over at UT Southwestern, and currently actually using the lipid nanoparticles, particularly designed for the Moderna vaccine, using this workflow. And we have some very, very promising preliminary data that shows that if you have the lipid nanoparticle sitting at room temperature for approximately 7 days, it'll have an increment in size. That means that the drug encapsulated within this lipid nanoparticle could be affected. Whereas if you keep it inside of the sieve or in this exoskeleton, it will preserve its size. Again, for 7 days. We only did structural characterization. We have not done more in-depth characterization of the material encapsulated within. But again, this is very, very promising results that we hope to follow up and eventually have even a more descriptive answer.
I think actually that to directly answer your question, we did a test like this where we put the proteoliposome formulation, so these very delicate membrane transporters in the ZIF, and compared it to refrigeration and how long they would last in the refrigerator. It was Sam or Nishi, you did this, right?
Yeah, we did that experiment. So even when they are like stored in the cold, within some time they tend to like deteriorate with their function. Like when I talk about the proteoliposome, so when they were encapsulated, the protection was more with the encapsulation. So we haven't done with the vaccines, but for the liposomes, it was the encapsulation was protecting it for a longer time.
Yeah, and if I may add on that, just to give an idea, I think that for that particular set of experiment, you are going from a stability that is lasting only a few hours at 4 degrees refrigerated for this system to a stability at even higher temperature that is lasting months. So it's actually a pretty remarkable difference.
Thank you. We do have a couple of additional questions related to the exoskeleton. They are as follows. Can the exoskeleton be used to stabilize vaccines other than COVID-19? And does the exoskeleton change the way the vaccine actually works in our body systems?
Yes. So we have looked at stabilizing whole cell bacterial vaccines, which is collaborative work that we're doing with the Danisco lab. So actually, we had a Research 411 on this a while back, where we were looking at creating whole cell vaccines using whole cell vaccines. So essentially dead bacteria that look exactly like the live version and trick the body into making strong antibodies against them. Our groups together are exploring other areas, including vaccines and tuberculosis and stuff like that. The second part of your question is, so there's two answers to that. One is if you remove the shell from the vaccine, so it becomes the same thing that the pharmaceutical company made. It doesn't change it at all. It just protects it. So it's like wrapping it in a blanket, and it gets where it comes and you take the blanket off, and it's identical. So we would not expect that to change anything. But if you leave it on, which you can, and do the same injection, what happens is it actually the shell dissolves very slowly. So it's it's keeping with our food theme. It's like an M&M, right? So the exterior shell keeps the warm, chocolatey, gooey layer all safe, but that's not to say that the shell can't also melt eventually. And in the body, the organic salts or inorganic salts that we have naturally in our body actually will pull the shell apart. And then that will cause the material, the vaccine, to come out. And we think that this is very beneficial, and hopefully we'll have some a paper on this very soon. But just to give you a preview, the way your body works in processing infections is you get an infection. It's not like in a vaccine. Like, you don't get an infection in your arm that then drains out within a couple hours. It's like, that's how a vaccine works. You go to the doctor and they inject it in your arm or in your kid's butt, and it stays there for a little while and it's gone. That's not how a real infection works, and that's why vaccines require multiple injections to keep the immune system like, oh, this is actually a problem. I should do something about this. And it's really just a timing thing, because the immune system just has to take its time. Because if the immune system reacted to everything it saw that it didn't immediately recognize, you'd be sick all the time. You would kill you. So the immune system has its function. It needs to take its time before it really fights an infection. And so the slow releasing of vaccine, actually what it does is it better mimics how the body actually deals with an infection. It slowly, over a very long period of time, allows the immune system to train the cells, and these are B cells that go off and they make the antibodies that I think we're all familiar with. These antibodies that then stick really tightly onto the virus, like COVID-19, they stick really tightly on the surface. And when they get covered in these antibodies, they can't get into cells and then they don't work anymore, right? But what this does is it gives the body a lot of time to sort of make these antibodies and make them even stickier. And you make them even more specific for COVID or tuberculosis or E. coli. And that means that you are even better, you're even more protected. And it also helps create the cells that are responsible for remembering the antigens for a lot longer. And so those cells, they go and they live in your body for a very long time, and they can be reactivated immediately once they see the infection again. Those cells are specifically trained to be activated instantaneously and start responding. But you have to make them from the initial infection, and that sort of has to follow a prescripted response. So yes, I think this research has two divergent areas. One is sort of the immediate practical effect, where we can sort of cut out the cold chain and then produce the exact same therapeutic, which is great from like an FDA standpoint, where this isn't an actual formulation. This is just something that we can put in, and then the stuff the patient gets is identical to the approved drug, or an entirely new formulation that slowly releases and promotes what are called germinal centers, and these centers are inside the lymph nodes and the other lymph tissue, because that's where your immune system really does its job, is in the lymph tissue, and it ultimately creates these really wicked strong antibodies. And so there's two different directions that we see, and our groups are kind of working on both right now.
Excellent. Thank you very much, Dr. Gaston Smith. There is one question in the queue. It is directed to Fabian, and I'll do my best to pronounce everything appropriately. This question again is for Fabian, and it is based on the 2021 Nature Pub. What is the pore size of the zeolite? What is the typical diameter of a proteoliposome? And so therefore, is the proteoliposome built inside in the zeolite, or is the zeolite built around the proteoliposome?
Okay, so first question, what is the pore size of this material? We measured porosity, and we came to the conclusion that whenever the proteoliposome is covered with this exoskeleton, the exoskeleton itself is not porous. In terms of the diameter of the proteoliposome, we did some experiments, some physical characterization of these materials, and they range anything between 150 to 250 nanometers in diameter. The last question, if I remember correctly, does this proteoliposome build on top of the sieve, or is it the other way around? So very simplistically, in very simplistic terms, you have the proteoliposome sitting in solution. And then the SIF basically covers it. Again, thinking of the Tony Stark, the Iron Man example, is just about putting the suit, doing your job, saving the world, taking the suit off, and being a normal human being, if that kind of makes sense.
Absolutely. Thank you. Well, our program is actually coming to a close. So Dr. Gaston Smith, Dr. Meloni, Fabian, Nisansala, and Samira, thank you for being on our program today to share valuable information with our community. Audience members, please consider attending additional Office of Research programming. Additional information related to other Office of Research events can be found visiting research.utdallas.edu. Thank you.